As I've already said, transcription is the most important level of regulation of gene expression. But in eukaryotes, there are many more levels of regulation. Here are a few of them. When it comes to transcription, there are two main aspects of regulation: chromatin structure, which generally determines which genes are accessible to transcription, and transcription factors, which are proteins that bind DNA and turn on transcription. These aspects are not independent of one another. Chromatin structure determines which genes are accessible to transcription factors, and transcription factors influence chromatin structure. We've already talked about splicing of eukaryotic mRNA. Here I would like to emphasize that it is a point of regulation. When a pre-mRNA is spliced, exons will be included in some cases and excluded in others. This is called alternative splicing, and it changes the protein that is expressed. Thus, a single gene can encode multiple proteins. In some cases, hundreds of different proteins arise from a single gene. MicroRNAs, or miRNA, are short RNAs encoded in the genome, and they affect gene expression by blocking translation of mRNA or by leading to the degradation of mRNA, which of course means it can't be translated. There are other levels of regulation as well. For example, the stability of a protein, how quickly it gets degraded, can be regulated. When proteins are first synthesized, they are often processed to their active form, another level of regulation. The efficiency with which an mRNA is translated and the efficiency of mRNA export from the nucleus are also regulated. There are other points of regulation as well, but we'll start with transcription and the two main aspects of transcriptional regulation in eukaryotes. We're accustomed to seeing DNA as a simple double helix, as on the right side of the figure. In fact, DNA in eukaryotic cells exists as chromatin, which is a complex of DNA and protein. For the sake of simplicity, let's say that chromatin is 50% DNA and 50% protein, as we've previously discussed. The basic unit of chromatin is the nucleosome, which is 147 base pairs of DNA wrapped around 8 histone proteins. Double helical DNA is 2 nanometers across, and it wraps around histone proteins to form a nucleosome, which is 10 nanometers across. Note the beads on a string appearance of chromatin. This is the open and potentially transcriptionally active form. The nucleosomes undergo coiling and looping to give rise to 30 and 300 nanometer chromatin structures. These represent the typical levels of DNA compaction in the cell. DNA compaction is necessary because the total length of DNA in a cell far exceeds the size of the cell. If we take this further, we get to an extreme level of DNA compaction, the pairs of sister chromatids that are visible in mitotic cells. The most extreme example of DNA compaction is the single X chromosome that gets inactivated in mammalian cells that have 2 X chromosomes, that is, in female cells. There's more detail about X chromosome inactivation in your textbook. For now, suffice it to say that very few genes are transcribed from the highly compacted, inactivated X chromosome. Which of the 2 X chromosomes gets inactivated is a matter of chance and is different from cell to cell. Other than the inactivated X chromosome, chromosomes are not usually in these extreme states of compaction. Chromatin is generally in the beads on a string form or a more compacted form. In general, the more compact the DNA is, the less expressed or transcriptionally active it is. The degree of compaction is a result of chemical modification of the DNA itself, methylation, and chemical modification of histones, methylation and acetylation. These patterns of modification are heritable. That is, they're passed from one cell generation to the next as a cell divides. One chromosome can contain thousands of genes, but the degree of compaction along that chromosome varies, such that some of those genes can be expressed and others not expressed. How tightly chromatin is packed determines whether the gene it contains are available for transcription. This partly explains tissue-specific gene expression. A given cell only expresses a fraction of all the genes in the genome. The rest are tied up in a tightly packed DNA structure where they are inaccessible to RNA polymerase and transcription factors. The packing of DNA can be heritable, so that when a differentiated cell divides, it inherits the state of DNA, which in turn determines which genes are expressed. When the liver regenerates, for example, it does so by cell division. As cell divides, they inherit the state of DNA packing and therefore the pattern of gene expression. This pattern of gene expression determines the cell to be a liver cell, so that liver cells give rise to more liver cells. Another way of saying this is that the new cell has a memory of where it comes from. Note that the new cell has inherited information in the form of DNA structure, in addition to the sequence of nucleotides of genes, which is the typical route of inheritance. When information is passed on by means other than DNA sequence, we call it epigenetics as opposed to just genetics. Compaction of DNA and the chemical modifications that drive it are also reversible. So while many of the genes in a cell can be turned off by a highly compacted DNA structure, that chromatin structure can be undone to turn genes on. We'll come back to this later when we talk about reproductive cloning. Here are the take-home lessons from our brief overview of the effects of DNA compaction, or chromatin structure, or epigenetics, on gene expression. The tighter the DNA is packed, the less available it is for transcription. The degree of compaction, in turn, is determined by chemical modification of histones and DNA. Information about the state of DNA is heritable and is a form of epigenetics. This also means that this kind of epigenetics plays out in terms of how it affects gene expression. X chromosome inactivation is an extreme example, where almost the entire X chromosome is transcriptionally inactive. Chromatin structure determines which genes are available for transcription, but it is the action of transcription factors that determines whether or not a gene will be transcribed. Think of chromatin structure as a coarse adjustment of transcription and transcription factors as fine-tuning. The most important step in controlling a gene is whether or not transcription begins, initiation. Initiation of transcription, in turn, is determined by binding of RNA polymerase at the promoter. This is similar to regulation in bacteria. As is always the case, though, the situation is more complex in eukaryotes. The important thing that they have in common is that transcription is regulated by protein-DNA interaction. Before we go any further, let's learn or review some terms. Elements are DNA sequences to which proteins bind, and the proteins that bind them are called transcription factors. The gene to be transcribed is depicted in blue. The promoter, shown in yellow, is the sequence immediately upstream of gene where RNA polymerase, shown in green, binds. When I say RNA polymerase, I'm really talking about RNA polymerase plus many other proteins that help RNA bind DNA and carry out transcription. Enhancers, shown in red, are distal elements, DNA sequences to which proteins bind, that regulate RNA polymerase binding at the promoter, and thus transcription initiation. Note that enhancers can be near or very far from the genes they control. Transcription factors, shown in purple, bind to enhancers and enhance RNA polymerase binding at the promoter, and thereby activate transcription. Because the RNA polymerase and its associated proteins are always present, it is the presence of transcription factors that determines whether a gene is on or off. I've described a situation in which transcription factors activate transcription. Transcription factors can also inhibit transcription by blocking RNA polymerase binding at the promoter. But activators are more important because most genes in humans are off by default. Let's put it all together in panel B. Transcription factors bind to enhancers, which may be far from the gene they regulate. The transcription factors influence RNA polymerase binding at the promoter. Because enhancers can be far away, their interaction with the promoter involves DNA looping or bending, which is aided by still other proteins. Transcription factors then interact with RNA polymerase and its associated proteins via mediator proteins, which don't bind DNA. Once the complex is assembled, transcription can begin. Note that gene A is controlled by one set of transcription factors, while gene B is controlled by a different but overlapping set of transcription factors. Thus, the set of transcription factors in any cell determines which genes will be expressed. I've presented chromatin structure and transcription factors separately, but don't get the wrong impression. Chromatin structure and transcription factors do not act independently of one another. Rather, they are part of a complex interplay whereby chromatin structure affects transcription factor binding and transcription factors affect chromatin structure. In this slide, I've tried to combine the effects of both chromatin structure and transcription factors. Let's start with chromatin and the basic unit of chromatin structure, the nucleosome, which is 147 base pairs of DNA wrapped around an octamer of histone proteins. Here you can see a stretch of DNA contained in several nucleosomes. As you know, the chromatin can be much more compact than what's shown here. Embedded within this chromatin structure is a gene and its regulatory elements. The protein coding gene in green, the promoter in orange, and enhancers in purple. This is not necessarily drawn to scale. The 8 nucleosomes shown here span about 1,500 base pairs, but a gene plus its controlling elements can span several thousand base pairs. At the top, you can see that the gene and these elements are tied up in chromatin structure and are inaccessible to transcription factors or RNA polymerase. This is a closed chromatin structure, also called heterochromatin, and is generally associated with inactive genes, or genes that are off. Note that it need not be as depicted here. If just the enhancers are inaccessible, that's enough to silence the gene. In any case, you can see that this gene is off due to chromatin structure. As I've already said, chromatin structure is reversible. But whether it's reversible or not, you can imagine another cell in another tissue in which the chromatin structure is more open. That is, the DNA is not so tied up in nucleosomes, like the example on the bottom. This open structure is also called euchromatin. The difference is these DNA modifications that we talked about before. Methylated DNA is associated with closed chromatin, and acetylated histone is associated with open chromatin. As you can see, transcription factors can now bind to activate transcription. The gene is on and the encoded protein is expressed. What you don't see, but you saw on the previous slide, is that these transcription factors interact with proteins at the promoter, and this interaction may involve looping of DNA. The slide illustrates nicely the accessibility of DNA and chromatin to transcription factors, but it's a two-way street. Transcription factors affect chromatin structure. Transcription factors that activate transcription, which is most of them, may have intrinsic histone acetylase activity, or they may recruit proteins with histone acetylase activity. The reverse is also true in that negatively acting transcription factors may recruit proteins with histone deacetylase activity. That wraps it up for transcriptional regulation. Now let's look at post-transcriptional levels of regulation. We've learned that most eukaryotic mRNAs are spliced. Splicing can also be a source of gene regulation because it may occur in different ways in different situations. Here's an example. Imagine a very simple situation in which a gene contains 4 exons and 3 introns. It gives rise to a pre-mRNA with the same 4 exons and 3 introns. The mature mRNA, however, only contains a subset of those 4 exons. One mature mRNA contains exons 1, 2, and 3, and the other contains exons 1, 3, and 4. In this case, we say that exons 2 and 4 are alternatively spliced. Sometimes they're included and sometimes they're not. You can imagine another situation in which the mature mRNA contains all 4 exons. It should be clear to you that these different mRNAs encode different proteins. Thus, the proteins produced depend on the splicing pattern, and alternative splicing is a form of regulation of gene expression. The splicing pattern can vary from cell to cell to generate proteins that are only expressed in specific tissues. Or splicing can be influenced by hormonal or environmental factors. As I said, this is a simplified case. The average human gene contains 9 exons, and most genes undergo alternative splicing. The approximately 20,000 genes in the human genome, therefore, can produce a much larger number of proteins. In 2006, Andrew Fire and Craig Mello won the Nobel Prize in Physiology or Medicine for their discovery and elucidation of RNA interference, RNAi, a hitherto unknown mechanism of gene regulation. RNAi is a fundamentally important natural mode of gene regulation and an important tool for biotechnology. There are two general ways to achieve RNA interference, via microRNA, miRNA, or small interfering RNA, siRNA. They are both short molecules, about 22 nucleotides in length, that act on mRNA. They differ in their source. MicroRNAs are encoded in the genome, just like the mRNA, tRNA, and rRNA that we've already learned about. siRNAs come from outside the cell, from viruses, for example, or they could be human-made and artificially introduced into cells. They both bind the same complex, RNA-induced silencing complex, RISC, and direct RISC to a target mRNA. A target mRNA is one to which the microRNA or siRNA is complementary. That is, base pairs form between the si or miRNA and the target mRNA. RISC can then act in either of two ways, depending on the degree of complementarity. siRNA is generally complementary to the target mRNA across its entire 22 nucleotides, in which case the RISC will cut and thereby inactivate the mRNA. If the complementarity stretches across just 9 nucleotides or so, RISC remains bound to the target mRNA and blocks translation. As a tool for biotechnology, siRNA has an unparalleled ability to block gene expression, which makes it useful in scientific discovery and in therapeutic applications. For example, a recently approved drug, inclisiran, note that it almost contains siRNA in its name, which goes by the trade name Leqvio, is an siRNA that blocks the production of a protein in the liver. Without going deep into the biology, inclisiran blocks a particular protein and thereby makes the liver better able to remove cholesterol from the blood, thus lowering blood cholesterol and reducing the risk of heart disease. Our next topic in regulation of gene expression is reproductive cloning. Cloning has several meanings. Gene or molecular cloning means to copy a gene from its natural environment and put it into a form where we can manipulate and alter it. For example, the human gene for insulin was cloned into bacteria, so the bacteria would make insulin that we then use to treat diabetics. Another kind of cloning is reproductive cloning or organismal cloning. This is nothing new. If you've done any gardening, you've probably cloned a plant. When you grow a plant from a seed, that's not cloning. That's sexual reproduction. If, however, you take a cutting or a bud from a plant and grow a new plant from it, that's asexual reproduction or cloning. And it works well in many plants, like the succulents that thrive in the San Diego area. That's exactly what's shown here. This is a sago palm, a cycad, which, like conifers or pine trees, are plants that reproduce by seeds but do not flower. They can also reproduce asexually. They're native to Southeast Asia, but they grow well in San Diego, and you see a lot of them around. As they grow, they send off these buds. There are several here, one of which is quite large. These buds can be removed and potted until they grow on their own. That's exactly what I did here. This is the parental tree, and this is a bud that was removed from it, which is now growing on its own. This plant is a clone of the parental plant. While cloning in plants and some simple animals is relatively easy, Dolly the sheep is an example of cloning of a large animal. She's not the first animal to be cloned, but she is the first animal to be cloned from a somatic cell. She's been immortalized by taxidermy and now resides at the Scottish National Museum. Dolly was born July 5, 1996, but she wasn't born in the usual way. Rather, she was cloned by a technique called nuclear transfer. Nuclear transfer starts with two cells, an egg from a female, which is what would normally be fertilized and develop into an embryo, and a somatic cell from the donor. A somatic cell is one that is not a sex cell. Sex cells exist in the gonads, ovary or testes, and give rise to gametes. Somatic cells are everything else. That's the great breakthrough with Dolly. She was cloned from an ordinary cell of the body. In this case, the somatic cell was from the mammary gland or breast tissue, hence the name Dolly, after Dolly Parton. This procedure starts with enucleation of the egg cell, that is, removal of the nucleus. You now have a cell that would normally develop into an embryo and a fetus, but is without a nucleus, so it doesn't have the instructions to develop. This is where nuclear transfer comes in. Those instructions are provided by the nucleus of another cell, in this case, the aforementioned mammary cell. The nucleus of the somatic cell is transferred into the enucleated egg. The newly reprogrammed cell is then stimulated to develop, and with luck, it will develop into an embryo, which then gets implanted into a third sheep, a surrogate mother, who then carries the offspring to term with the birth of a live lamb, Dolly. In a sense, Dolly has three mothers: the one who provided the egg, the one who donated the nucleus, and the one who carried the pregnancy. It is the donor of the nucleus, however, that was cloned, because she provided the genetic material and all the genes that Dolly has. So what is Dolly? A Scottish blackface or a Finn Dorset? Pause the video and answer that question. The process is very inefficient. The scientists who cloned Dolly did 277 nuclear transfers and implanted 29 embryos to get just one live birth. Nonetheless, the procedure has been duplicated in many mammals, including mice, cats, mules, rabbits, monkeys, pigs, deer, horses, bulls, and dogs. The techniques have improved, but it's still an inefficient process with about 10% success rate, and it's very expensive and time-consuming. Given these obstacles, what are the practical applications of cloning? There are applications in agriculture. If you have an especially productive cow or a rapidly growing hog, you may want to clone it and capitalize on its productivity. There are also medical applications. Scientists have engineered and cloned a pig whose immune system doesn't reject transplanted organs and tissues. Such pigs could be used to grow human organs for transplant patients. The inefficiency and expense of these procedures limit their applicability. If, however, you have the money to burn, there are more things you can do. Barbra Streisand, the world-famous singer and actor, had her pet dog cloned, and now she has two more of a pet that passed away years ago. Perhaps the most promising application is in conservation biology. Przewalski's horse, Equus ferus, which is a native to Central Asia, is a good example. It had gone extinct in the wild, but was kept alive in zoos around the world, including the San Diego Zoo and Wild Animal Park. It has been reintroduced to its natural habitat, but all of the horses that now exist in the wild are descendants of just 12 horses, and even that small amount of genetic diversity is declining. Cloning was done in an effort to conserve this species. Starting with donor cells that were frozen at the San Diego Zoo in the 1980s from a horse that has long since passed away, scientists were able to reproduce 2 live offspring. The hope is that they can be introduced into the wild and increase the genetic diversity of the species. Similar efforts for other endangered species are underway. This approach is not without its critics. They argue that clones, by their very nature, don't really increase genetic diversity, and that the tremendous money and effort put toward cloning is better spent on traditional conservation efforts, like habitat preservation. The real value of Dolly and cloning, however, is not in their practical applications. Rather, it's in what they teach us about development. Earlier I said the state of chromatin structure determine what genes can be expressed. As humans develop, cells differentiate into the many cells that make up an adult. Differentiation depends on successive generations of cells inheriting the chromatin structure, epigenetics, necessary for the differentiated cells. Dolly proves that this differentiated state is not fixed, but is reversible. To borrow a quote from the Campbell textbook, Dolly demonstrated that the differentiation of animals is achieved by changes in gene expression rather than by permanent changes in the genes themselves.